Welcome back. In this video on muscle physiology, we'll look at the very basics and an introduction to muscle metabolism. So we'll start with the question, where does the energy to fuel muscle contractions come from? So we'll look at ATP production. So we mentioned earlier, adenosine triphosphate or ATP is known as the energy currency of the cell. And what we have at rest is a small supply of ATP is stored within the muscle. And this is effectively only enough for a few muscle twitches or a few muscle contractions. The rest of the ATP must be produced by metabolism of energy sources such as food. So when we metabolize food, when we break that food down into its smaller components, we use the energy from that food to combine adenosine diphosphate and inorganic phosphate into ATP. And that energy that's locked within there, we release it when we break down ATP again within the muscle. And then we use that to fuel the cross bridge as we discussed in the previous videos. There are three systems predominantly which the muscle generates ATP through. Is the phosphagen system, anaerobic glycolysis, and oxidative phosphorylation. And we'll go into those in a little bit of detail now. So the phosphagen system is based on phosphocreatine. So you have creatine plus an inorganic phosphate plus energy created within that. That energy is used to bind adenosine diphosphate and inorganic phosphate to ATP. This provides enough ATP for about 3 to 6 seconds of maximal contractions, and it's important during sports and activities which rely on explosive power. So think of sports which are very, very short in duration. They might be throwing events or jumping events or something like that, where you have a very short duration of maximal contraction, so power type sports. That's where the phosphagen system is predominantly used. After that, ATP would have to be generated from other sources. The second system is glycolysis, and what we're going to first look at is anaerobic glycolysis. So this is glycolysis where oxygen is not readily available. And glycolysis is the breakdown of glucose or glycogen to release enough energy to form ATP. So glucose or glycogen are broken down into G6P, then to pyruvate, and in the absence of oxygen, ATP is generated with lactate being generated as a byproduct. So that energy that's released from that reaction is used to combine ADP and inorganic phosphate to produce ATP. Oxidative phosphorylation then is when oxygen is available. So that's the breakdown of glucose or glycogen in this instance here into ATP. So as we've seen in the previous slide, if oxygen is not available, pyruvate is converted to lactate, and the energy from that reaction is used to form ATP. However, if oxygen is available, pyruvate is converted to acetyl-CoA and enters something called the TCA or Krebs cycle, and then into the electron transport chain. Now the benefit of when oxygen is available is we can create a lot more ATP. Okay, so we can create about 36 ATP from one molecule of glucose, 37 from a molecule of glycogen, and if we metabolize fat sources, we can attain up to 129 ATP from a similar amount of fat, because fat is quite energy dense. However, we can't. We need oxygen available to actually metabolize fat in this way and get ATP from it. We can't do that in anaerobic fashion. So a quick summary or review slide here. Creatine phosphate is broken down into creatine and inorganic phosphate, and the energy released from that creates ATP. Glucose is broken down through glycolysis. In the absence of oxygen, lactic acid is produced or lactate is produced as a byproduct, and that is used to produce ATP or to form ATP. If oxygen is available, if oxygen is an input here, we use oxidative phosphorylation. to create ATP, and we get much more bang for our buck. We get a lot more ATP generated if oxygen is available. We can also, if oxygen is available, use the energy from amino acids, which come from proteins, and fatty acids, which come from fat sources, to again generate ATP. Here's just some of the characteristics of different types of muscle fibers. So we have our slow oxidative type 1 fibers. These are fast oxidative fibers like our type 2A and type 2B, and then these are our type 2X fast glycolytic fibers. So here we have the primary source of ATP production in slow oxidative fibers, as they're named, oxidative phosphorylation. In fast oxidative, it's still oxidative phosphorylation with some glycolysis, and then in fast glycolytic, the ATP is produced in the absence of oxygen. They have different amounts of mitochondria, which is the powerhouse of the cell where this ATP is produced. Many mitochondria in slow and fast oxidative fibers, and much less in glycolytic fibers. The capillaries or blood supply is much greater to slow oxidative fibers than it is to glycolytic fibers. What we see is that the glycolytic enzyme activity will be much lower in your oxidative fibers and much higher in your glycolytic fibers, and that would make complete sense. Similarly, the glycogen content is low in these oxidative fibers, but higher as we go towards more glycolytic fibers. The rate of fatigue is lower in slow type 1 type fibers and higher in type 2 type fibers, and we spoke about that in the previous video. The fiber diameter is much smaller in our type 1 fibers and greater in our glycolytic fibers, and that's true for the motor unit size and the size of the motor neuron innervating the fiber for both slow and fast glycolytic fibers. Okay, just something to consider for yourself. Which energy systems would these athletes utilize in their event?